title of Bioremediation of Hydroponic Greenhouse Effluent and Bioagrochemical Applications with Microalgae.
Hi, first I would like to express my sincere sadness and despair about the tragedy happening in the southern eastern part of Turkey, and all my prayers are with the people in there. And I would like to thank the organizers for gathering engineers in Istanbul. Today I would like to introduce you the studies that we are doing in photomicrobes group on bioremediation of hydroponic greenhouse effluent, and also bioagrochemical applications with microalgae. Baltic Sea knows one of the most polluted water bodies around the world, and it suffers from eutrophication, and also algae bloom news are not a surprise for the summertime, especially warm summers in Finland. And according to the Finland Helcom report, 46% of the nitrogen inputs in rivers and 35% of the total loads of phosphorus in Baltic Sea caused by the agricultural wastewaters. And as you can guess, it is because of the excess use of the fertilizers, synthetic fertilizers, and if we take a magnification to the amount of the money that Finland spends for the fertilizers per year is 250 million euro. To fertilizers, soil improvers, and also plant protection products. But with the awareness of the consumers, now there is a demand for more sustainable foods and also with the lower impact of the environment. Agriculture and Finland, if you think about it, if you think about the geographical location and climate conditions, due to harsh cold season, you can guess that year-round cultivation of the crops mostly depend on the greenhouses. And as you can see on the slide, that tomatoes, cucumbers, and ornamental plants are the crops that mostly produce in the greenhouse production. Greenhouses as products. And in the last decade, hydroponic greenhouses are also trendy because of, as Marianna mentioned, because of the advantages within the greenhouse farmers. However, as we have talked, there is no strict regulations for the greenhouse wastewater discharge yet in Finland, even we are following the European regulation. As Marianna mentioned, it is depending on the country, yes, that's right, but as far as we know from the Greenhouse Product Association in Finland, that it is also depending on the municipalities. You can see in here chemical characteristics of greenhouse, hydroponic greenhouse effluent from a commercial cucumber producer close to Turku. You can see that it is from hydroponic one, so soilless. Turbidity is quite nice to let grow the algae, and also it is really nutrient rich if you take a look to the nitrogen and phosphorus amounts. But even some of the growers, they prefer semi-closed circulation. What does it mean? Maybe 40% of the water is recirculated in the hydroponic greenhouses. If you think about Finland and think about also temperature, you know, heating also with the lights, you can think that they don't want to risk the crop yield. So that's the reason also excess fertilization and lower amount of recirculation is also understandable. And they don't want to also risk their crops with some of the organisms like some of the fungi, for example, especially for the cucumber. So that's the reason they are avoiding, let's say, that recirculate that much their water. So after these problems, what is our suggestion? A green solution, microalgae, and we come up with the integrative algae and greenhouse production. We would like to use already established infrastructure of the greenhouses in Finland, fully controlled, automatic controlled, and then use their effluents to grow the algae, produce clean water, produce a value product as algae biomass from the West, and then with the biorefinery concept, turn into value with the high value products, low value products. And of course, for the clean water, let's start with the bioremediation studies. We started with screening, and for screening, we have a Nordic culture collection. You can look for the details on the webpage, nordacoa.fi. Nordacoa is a Nordic consortium, and it merged 3 existing Nordic cultures from Sweden, Norway, and also Umeå, Sweden. And it consists around 3,000 species, not only from Nordic countries, but also from all over the world. And we have, of course, even we have 3,000 species, we haven't done 3,000 species. We started with the strains that promising, and we are thinking that with the fast-growing biomass accumulation ones, and we have tried in the beginning 13 species in the multi-well plates, and we would like to see how they are reacting to the wastewater, and then also if wastewater needs any adjustment. And we have seen that most of the strains were promising from the growth rate, and we don't need any adjustment for the hydroponic greenhouse to let the algae grow. And from the strains, we would like to continue with the Synedesmus, which our lab is familiar with, and also we would like to continue with the Tetradesmus, show in the growth studies one of the good results from the OD results. But of course, when we did the screening test, we have seen that also domestic algae is growing in the wastewater, so we would like to do a pre-treatment. In the bench scale, we gave a try to the filtration, but we always think about easiness of the operation in the pilot scale also. So we tried the filtration, a coarse filtration, like filter sterile 0.2, 0.5 kind of filtration, and also chemical bleach plus neutralization. And we have seen that filtration is working okay, so we continue with the filtration for the pilot scale studies. We have a paper that already published, so with the Synedesmus strain, after the lab scale studies, we passed to pilot scale studies in a greenhouse environment with the commercial cucumber greenhouse effluent. We used the Synedesmus strain, and we did our, we use a tubular photoreactor you can see on the photo, and as you can see, we did more than 1 month of continuous turbidostat operation. And as you can see in here, nitrogen and phosphorus values varied between 240 up to 300, and phosphorus values varied between 50 to 30. It is depending on the season, it is depending on the crops, age, etc. You can think like that. And we have used halogen lights, and we mimic the cucumber production photoperiod light intensities, and we have seen that with this, also I would like to add that pH was adjusted to 7.5 with the CO2 addition, and we have seen that we have promising removal for the phosphate depending on the different parameters. However, nitrogen removal is moderate. So, from these studies, where you can see the details in this paper, we have decided that we need to enhance the nitrogen removal, and how we thought about that. And we know that also in the cucumber production, they use LED lights, so we would like to give a try with the LED lights, and also Tetradesmus strain, we would like to give a try to to get more biomass accumulation after seeing synedesmus is okay in the pilot scale studies. And we would like to also check this time in the batch scale the nutrient dynamics. With the LED lights around 300 micromole, and also with the additional natural light, because we are in a greenhouse, glass greenhouse compartment, with the use of, again, cucumber greenhouse effluent, we started to grow our algae. And this time, comparison to the synedesmus strain, we have seen that after the 4-5 days, our strain accumulates more biomass, which is promising, which we expected. But after the nitrogen removal also happened, it continued to grow, which from that part, I would like to say that we were thinking that there is going to be maybe phosphorus addition needed. However, we didn't. And we have to have an insight, we have followed the batch cultivation till to the stationary phase. And we have seen that also on the third day of the cultivation, 100% of phosphorus removal we have achieved. And on the day 13, we were able to reach the European legislation values, and 15th day, we have reached 100% of removal for the nitrogen, and we had around 5 grams per liter maximum dry weight. In here, I would like to take your attention that this first trial done in November, which means that 6-7 hours of daylight. And as you can see, this blue shows the outside temperatures. Yes, this turquoise, let's say, and blue shows the temperatures inside the greenhouse compartment, and then the red shows the temperatures in the PBR. And this was our first trial, and following the synedesmus trials in March, we would like to give a try in a longer daylight period, like 12 hours, and with more solar radiation time period, and we did trials in March again to see how the strain will react. Then in March we have done our trials. You can see the difference in here with the temperatures and outside, inside. And more or less, we are always starting from the same inoculation ratio, and you should think that also even the greenhouse water having some variances in the nitrogen and phosphorus, it is always between 240 to 50. Okay, I have time. So then, if you take a look to the graphs, you can see that there is a huge difference in the solar radiation for the March period, which is as expected. And we have seen that the nitrogen removal acceleration is faster in March period. So we would like to dig into the details of how the curve of the dry weight and nitrogen removal is going on. I forgot to say, of course, in the trial 2, we have reached, as in the trial 1, on the day 3 for the phosphorus removal, but this time faster removal was obtained for the nitrogen on the 8th day, 100%, and we have reached higher dry weights also. But in here, of course, we are making the experiments to have inside details for letting us to continue to the continuous system. Now let's take a look to first 8 days of the cultivation, which if we magnify, we will see that dry weight numbers of the both strains are more or less the same, following the same trend. However, not the nitrogen consumption. You can see a huge difference in the acceleration of the nitrogen removal in the second trial in March. And you can see that the LED lights are more or less the same. And in the first 8 days of the trial, PBR, inside the PBR, the temperature is more or less the same also. The difference is the solar radiation. So this means that light is more important than the temperature, which is as expected, and also you need to think that in March, culture gets like 12 hours of daylight, when in November it is like 6-7 hours. And when we take a look into the nitrogen removal uptake dynamics, we have seen that in trial 1, we have reached like 75 to 95 milligram per gram biomass removal per day for the batch cultivation. However, in trial 2, we really exceeded it, and it became like 125. And what this shows to us, this gives us enlightening values to let us to put our threshold values for the continuous system. We are aware of that. You can't wait for 10 days to remove the nitrogen. You can see the details of the studies in our paper. Now, also I would like to tell to you that we have taken also biomass samples in the beginning of the experiment when the phosphorus is all removed, and also at the end of the experiment. And we have seen as expected that in the exponential phase, we have more proteins, and we have more carbohydrates in the stationary phase in this nutrient, first nutrient replete, and after a while nutrient deplete condition. Now we have an idea about how we are able to reach clean water. So then we would like to create a pipeline for our bioagrochemical studies with the Nordic culture collection that we have, and we have started our biostimulant studies. We would like to check the microalgae with potential for the plant biostimulants. But this time I would like to say that these strains were grown in mostly like BG11 media, which also really close from the nitrogen content to wastewater greenhouse effluent, more than 200 milligram nitrogen per liter, and phosphorus ratio a little bit lower than the greenhouse effluent, but not low, as far as I remember, 7-8 milligram per liter. Then we have grown the microalgae in BG11, mimicking the greenhouse effluent, freeze-dry it, and we would like to try with 2 extraction systems to compare, and we would like to do a pipeline experimental setup, one with the model plant Arabidopsis, and to check the results of the model plants, we also did greenhouse lettuce experiments. For the extraction, we decided to do hydrolysis. Since we know that our strains have proteins and carbos, we decided for the hydrolysis, acid hydrolysis followed by ultrasonication, and wheat peeling followed by centrifugation. And from the results that we have obtained, of course, I don't need to say that we have tried different concentrations. We have done preliminary tests to reach also these concentrations. We have seen that Arabidopsis rooting bioassays showed that us hydrolyzed extracts from algae enhanced root growth, and some of them also increased the lateral root growth. And effects of the beet meal aqueous extracts are highly concentration dependent. We saw that some of the concentrations show some even inhibition, especially for the lateral roots. And to check these results with the greenhouse experiments, because it's in a way rapid screening, we passed to the greenhouse lettuce experiments, and the results were supportive. Hydrolyzed extracts increased the lettuce fresh weight around 12 to 15%, and wheat meal extracts, depending on the concentration, it increased the fresh weight of the lettuces, which means that our baseline experiments are okay to further go with the wastewater-grown microalgae biomass to compare their biostimulant effects. And now let's take a look to our biopesticide studies. Again, we have screened microalgae species from Nordica Nordic culture collection, 13, 15 strains, and we have chosen the plant pathogens relevant to the Finnish agriculture. You can see in the table the plant pathogens matching with the crops that affecting in Finland. And we have seen that some of the strains didn't show any effect, antifungal effect, antimicrobial effect to the chosen plant pathogens. But for example, 5-6 strains in here were promising. And Chlorella sorokiniana is one of the most promising one, especially for the, which I can't pronounce the name now, Phytophthora cacturum, let's say cacturum after now. So for the cacturum, it showed one of the best antifungal activity. So to check the extraction and concentration dose dependency, because we didn't do the optimization at the end, we decided to go with the Chlorella sorokiniana and then the cacturum plant pathogen to check how the things are going on. What we would like to see? We would like to see the growth inhibition of cacturum exposed to chlorella extracts, and we would like to follow 2 routes, dry and wet. And then we would like to check the different solvents, thinking about the polarity and non-polar and polar compounds. And then also we would like to see the different fractions, how it affects. So we tried the crude extract, soluble part, and the pellet. We did the antimicrobial tests on the plates, and then also we would like to check on strawberry plants, which the cacturum is affecting, how these extracts will work. We have tried, as you can see, wet, dry, acetone, and milieu, and with different concentrations of the microalgae biomass. Our results show that water-soluble compounds are affecting the antimicrobial effect of the cacturum from the sorokiniana extracts, and then also drying, enhancing the extraction. If you think about acetone, it's not a surprise, I think so. And then also with the extraction methods, which is not optimized, now we are a little bit depending on the concentration of the microalgae biomass. And with the most promising sorokiniana extract, which is the soluble part of the 100 gram per liter, as far as I remember, we have tried, we have dipped the detached strawberry leaves into the extracts, and as you can see in the figure, you can see that this is the milieu one, and this is the ones that with the algae extract, and those are the leaves that they are infected with the cacturum plant pathogen. So our results are promising from that side if you think that even we didn't optimize the extraction methods. So therefore, we have a baseline for our agrochemical studies. We will continue to work with wastewater-grown microalgae species, hopefully. So what we are gonna do next? RealM. As Marianna mentioned, we are pleased to take a part in RealM project, and we are gonna act for the bioremediation studies as a validation plant with our 1 meter cubic reactor with the greenhouse wastewater effluent in continuous mud turbidostat studies, and we are gonna also take a part in the bioagrochemical trials. Of course, this is a teamwork, no need to say, but nothing can be done without money, so we would like to thank to the funding agencies, and I would like to thank to all co-authors of the articles. but especially João for bioremediation studies, Martina for biopesticide studies, Eric for biostimulant studies, and of course to our team leader Yakut for leading us. And last but not least, to all Photomicro team for their support. Thank you. If you have any questions.
Thanks very much. Thanks, Emma, for a really very beautiful talk and very inspiring. So I've got a question. As I used to. I mean, if you want to ask a question, you're most welcome. I don't have to be the first one. Did you want to ask a question? Okay. Right. So, this lateral root stimulation, because you obviously check the effects of the microalgal extract as a biostimulant, biopesticide. So coming back to this biostimulation effect. So this lateral root stimulation, do you have any idea what might be responsible for it? Because in plants, it's obviously auxin mainly. Can it be any kind of specific enzyme that produces some sort of auxin equivalence in these algae? What would be the metabolite or molecular component that would be responsible?
Actually, there are hormones that already we know that affecting the plant for example, their root growth from the length.
Algae. What is the stimulant in algae that would?
Their hormones, like giving the gibberellin effect, like hormones, and then auxin kind of hormones that they are.
Sort of like
yes, they have also that. Yeah, and then also. So that's the reason also the algae, depending on, of course, also amino acids, if you degrade them into smaller molecules, let them to get from the plants, etc. But phytohormones, amino acids, and then also, even it is a little bit gonna expensive with the microalgae, carbohydrates also. So those are the effects I can tell that with microalgae.
This is great because, quite a, auxin, synthetic, lots of synthetic auxins have been obviously used to promote this lateral root growth, and you can do that without it.
So that's the trend also now in the microalgae with the high volume products for the biostimulants. Any more questions?
Ses? Thank you for the inspiring presentation. I want to ask, you did about like an organic pesticide, right? Algae pesticide.
Yes.
Yes. Did you check, right, without the pathogen, what this pesticide, what is the effect of this pesticide to the growth of the plant that you studied?
Yes, but you are using this biopesticide in the case of there is to stop the this plant pathogen effect on the whatever the crop you are. So if I'm right, you are asking that without infecting the leaves, did you check what this pesticide is doing? And so it is not becoming a pesticide then. So that's the reason it should be checked in another way, like if our extract is affecting already grown strawberry, in which way, which it was not in the target of our study, so we didn't do. But it might be done also if you would like to try this extract for other purposes after because we have done it after the plant became as a strawberry, not to lose the product. So it is not for enhancing the growth, it is for keeping the strawberries in good shape, not to waste your already grown strawberry plants, let's say like that.
And it doesn't affect, for example, it doesn't negatively affect the grown strawberry?
No, it is, we have done with the strawberry actually, with the strawberry itself, but we need to optimize it. Depending on the strawberry, because you don't know what we have in the strawberry, you need to check also what they have as pesticides or etc. When you are buying a strawberry from the market, it has also something. So we need to make a deeper analysis for that. But we have tried for the strawberry. Some of our results are promising, but it needs deeper research, let's say.
May I ask one more question?
Let's give. We can talk at poster time if you would like to, and then if another one would like to ask, I'm suddenly becoming like a teacher. Sorry, but
Thank you.
yes.
Thank you so much. Two quick practical questions. One is the light system very similar to the that used in the greenhouse? So
Yes.
you can use both for algae and the plants?
This was the idea.
That
Was the idea because halogens are already there. Only the difference with the LED lights are, depending on the greenhouse, they can have these head LED lights or they can have side LED lights, but we already know from the websites or literature that these LED lights also in Japan, Korea, also using for cucumber cultivation. It is a commercial LED also they use in the greenhouses. So this was the idea of it, mimicking greenhouse conditions.
Another practical question. Some crops do require a little more acidic, water. You got really good results in acidic hydro. Can you keep the pH or the pH goes back to neutral level?
That's a really good question. This was also considered and we have checked, but when you have the first done the stock extract, which is really concentrate, and when you start to dilute it, if you start from, think that 50 gram per liter with the hydrolysis, and when you come to 0.05, so then with that much water, your pH is not becoming that much acidic also. So, and also we have done the trials with neutralizing it, then depending on which OH, sodium OH or KOH you use, we have checked also with that, and since it is really trace amount, of course this is not affecting also, so yes, we should do the, of course, neutralization if it is that much acidic.
Thanks so much.
Thank you.
Thank you for the presentation, Dr. Sema Shirin. Now we have a video presentation of Dr. Luisa Guea. Then she will be connected to answer the questions. Luisa Guea is a chemical engineer with a Master of Science in Food Science and Technology and PhD in Biotechnology. She has 35 years of experience in microalgae, biotechnology, and coordinator of several national projects related to macroalgae. Luisa was vice chair and national delegate of the Coast Actions for 1408 EU Algae European Network for Algae Bioproducts and vice coordinator of Ibero-American Network Wastewater Treatment Project Renewal. She was also coordinator of bilateral project between Portugal and Serbia on the production of high-value products from novel marine macroalgae using green technologies. Luisa also coordinates bilateral project between Portugal and India focusing on combination of vertical wetlands, microalgae photobioreactor and microbial fuel cell for wastewater treatment in small pig production farms. Luisa is author or co-author of more than 110 international peer-reviewed papers and book chapters.
Hello, everyone. I'm very glad to be here in the Algae Nexus Conference. Thanks, Ebarat, for inviting me. I wish I would be with you, but because I really love Istanbul as well. But anyway, I will show my last work, Pig to Wheat, which is basically our motivation was the world population is growing, we know this, and it seems that in 2050 we need 60% more food, and agriculture will decrease more than 10% because of the climate change. The synthetic fertilizers, the emissions are great, are too much, 13% due to synthetic fertilizers. And the piggery wastewater is a huge problem in Europe because they are always increasing and generate highly polluted wastewaters. We have several problem backgrounds. How polluted are the sectors livestock and agriculture? A lot. The livestock is the emissions are around 15.5% of the total emissions of the greenhouse gases. And in terms of agriculture, as I told you before, 30% only for the synthetic fertilizers, but 7% for the manure management. So also another problem is how certain is our food. Also a lot, because agriculture has a global water footprint of 92%. It's huge. And 70% of the fresh water is to irrigate the agriculture. And for the increase of 60% of food production, we also need 40% of water. It's incredible numbers, and 1 kilo of pork requires 6,000 liters of water. Again, 1 kilo of wheat around 2,000 liters. And we have billions, almost 500 billion liters only in Europe for the pork sector. It was 2 years ago, but so maybe it is still increased. And what we have to have in mind is we have to reuse the water and recovery nutrients, absolutely. The overview of work is we start with the pig farming. With wastewaters, we want to cultivate the microalgae and treat the water at the same time. And then we produce the microalgae biomass and obtain the treated water. And after processing the biomass or not, we evaluated the potential in agriculture of this all biomass and the treated water. And again, these cereals could be used again to the pigs. The challenges, it's incredible, the color and the characteristics of the piggery wastewater. You see so dark and so high COD, total solid suspended, and the color. And again, horrible is that the ammonia is 10 times more than the tolerance of toxicity for the algae, and a big predominance of free ammonia in alkaline environments, and a very dark color and high turbidity, which is very difficult to the light, impossible to the light penetration, and of course the photosynthesis. However, lots of papers used the dilution to treat the water, but we don't want this strategy because you don't want to use fresh water to clean dirty water. And as you know, we have a lot of scarcity of water all over the world. You see here the numbers are incredible. So we don't want this strategy. What we did was test several pretreatments like electrocoagulation, ammonia stripping, constructed wetland, and photophantom. And from all of them, the best one, it was photophantom. Keep studying what the photophantom can be doing for our effluent. And we did this low pH, 120 minutes of UV, iron and hydrogen peroxide, and and then after neutralization and settling for 1 day, and we obtained this clean pre-treated supernatant and the concentrate. And we thought, what? We have here a problem. This concentrate will be a problem, but we had a nice final story about this concentrate. And in terms of concentration of the CODs, it was reduced a lot for the values that are permitted for the algae. also the TSS, the suspended solid, total suspended solids, at the color, and anyway the ammonia still very high. But then we evaluated the concentrate, and when there's a lot of minerals, we thought maybe it could be a good fertilizer or not, or will be a problem. And then after cultivated, we use the culture directly, or after harvesting by flocculation, electrocoagulation, and centrifugation, or the combination of flocculation with centrifugation and electrocoagulation with centrifugation. With these with these parameters, centrifugation we use 4000 G for 5 minutes, flocculation with ketosane 25 milligrams per liter, 1 minute fast, and then to start the flocks, and then 3 minutes very low to coagulate the flocks. And we also use the electrocoagulation with combined iron, magnesium, and zinc for 8 minutes. And then we use the biomass of these all different cultures, and also the supernatant. And we did a germination for 5 days and the plant growth for 6 months. But you also use the supernatant of the phytoplankton process and the concentrate. And then we think that we didn't do yet, but we will do the cereals, the wheat produced to feed again the pigs. The germination for 5 days, the control was distilled water, and the results are these. All the microalgae in green are better than the water. The water is 100%. And the supernatants didn't work at all. You see, the microalgae is the best. Mainly centrifugation and flocculation. And then we evaluate the wheat growth by tailoring and steam elongation for 82 days. And here we also use another control, a positive control, Oglan solution, which is a fertilizer solution. And as you see, the supernatants are very good in number of leaves and the number of tiles and also the length. And here we can see the photos. I'm not lying. Water is nothing on this side, on the left. The oval is okay, and all the supernatants are very good. From flocculation, from electrocoagulation, here from centrifugation, and the combination of flocculation and centrifugation, and electrocoagulation and centrifugation. Here we also saw a nice result, because we saw that the phytoplankton works, which is, instead of being a problem at the beginning, it's very good. They have very good results, very green wheat plant. Here the microalgae also is good. And then after 182 days, we evaluate the shoot growth. Again, the microalgae and the phytoplankton concentrates. And the number of crops, the same, the phytoplankton and the microalgae. The Oglan is always good because it's fertilizer. A good result is if it is similar as Oglan, then we avoid the synthetic. But we have with the phytoplankton concentrate even better. So it's a very good. As a conclusion, we can say that phytoplankton is a pre-treatment, very good step, because then the algae could grow in undiluted piggery wastewater, so we reduce the water footprint. The microalgae works as a germination enhancer. The microalgae achieves higher germination values than the control, so a biostimulant for germination related to the microalgae. The harvested supernatants are enriched irrigation water. The supernatants obtained after biomass harvesting are rich in nutrients, so we obtain a water enriched, which is also good. Phytophthora concentrates can be a biofertilizer. The phosphorus, iron, calcium, and potassium who are beneficial for the growth, and this allowed us towards zero waste from. Just an image of everything what I just said: reduce water footprint, reduce nutrient costs, reduce synthetic fertilizer demand. As we saw, the macroalgae would go directly to the feed or as a biofertilizer stimulant for the wheat. The treated water and the precipitate goes directly to biofertilizer to wheat and then to the pig. So it's really a process towards zero waste, which is very good. It's really a economy. My question is, can the tiny algae solve or fight all these problems that I mentioned at the beginning? In my opinion, they will not solve all the problems, but they will fight all, and they will greatly contribute for the solution. And about the future, who knows? We never know the future, but maybe a relaxing beer made by algae in the space. We know that the algae are already in space trips because they need something to clean the gases and the wastewaters. And maybe also we could walk in the nice light countries as Portugal with sun, and then we can eat and drink at the same time. And this is my team, my collaborators, and thank you. I also want to acknowledge the project that they were involved in this work. Thank you very much.
The connection was problematic, that's why she presented her topic with the video. If we will be connected well, we direct the questions. We need to check.
I think she isn't here. I think connection problem is continue.
We couldn't solve the problem, so we will direct your questions with the email. Thank you. Now we will give a break for the poster session. Thank you. Until we can meet at 15:30.
Oradan e-mail'i görsünler diye diyorsun. Tamam.
Geliyorlar hocam birkaç kişi daha.
Tamam. The fourth session, as you can see, we were attached to Dr. Lisa's email address. If you have question, you can direct your question with that email address. Thank you. Our fourth session will start with Dr. Mehmet Ali Küçüker from the İzmir Institute of Technology by the speech title of Investigation of Biofilm Structures of Chlorella vulgaris and Ethylea oleoabundans species on an anti-biofueling coating. The stage is yours. Kontrol edebilir misiniz hocam? Okay.
Hello, everyone. It's really difficult time to be here because of the earthquake, but we are sad. However, the professional life is going on, so we are here.
I would like to thank the whole organization here. First of all, I would like to show our last results from our project, which was related to investigation of biofilm structures of 2 different microalgae species on the anti-biofouling coating. So in the previous presentation, we observed there are main global issues which are listed like this: energy, water, food, and environment. So nowadays, researchers have focused on solving these problems. We always discuss with Professor Berat Haznedaroğlu. He always mentioned that we can find a solution for micro problems using microalgae. We know it's great potential. It has a lot of properties to solve this problem, such as the most efficient photosynthesis, high growth rate when we compare the ordinary foods. Independent of agricultural land, it can be grown in fresh, salt, and brackish waters. So it has a potential for carbon dioxide removal. And we utilize microalgae to treat wastewater. And also we can utilize microalgae to remove phosphorus and nitrogen from wastewater as well. Mainly cultivation system depends on the 2 categories. One of them is open pond system. You can see the right corner of the slides: raceway, pools, and ponds. And there are engineering systems, mainly photobioreactors, glass tubular photobioreactors, panels, flat panel photobioreactors, airlift photobioreactors, and column photobioreactors are mainly used. We mainly focus on the closed system because of the European microalgae cultivation depends on the closed system. Approximately more than 70% of the cultivation systems belongs to closed photobioreactors. So we focus on to increase the efficiency of closed system, mainly tubular systems. That was our projects, which was funded by the TÜBİTAK, and we aim to increase the performance of the tubular photobioreactors, and that will allow to produce of reactors with the high light transmissions, and we would like to decrease in biofilm generation on the surface of the bioreactors. And we improve and develop special coating materials using the osmosil, nanoparticles, silicons. So that kind of coating materials has properties under the surface waters, like superolympic conditions surface, but in the air, open air, it's super hydrophilic surface. So this is the main important part of the coating materials. And we would like to increase the productivity by 50% because of the less biofilm forming systems. And then we only utilize the water to clean the system. As you know, if you would like to cultivate different species, you have to clean up the reactors with some chemicals. And at the end of the day, you cannot utilize the water because it is wastewater. It is polluted by the chemicals. So we would like to reduce the use of chemicals for cleaning reactors by 100%. and then at the end of the day, we decrease the water footprint for cultivation of microalgae. And this study aims to investigate the ormosyl coating classes having anti-biofouling properties to overcome the biofilm attachments. So we use column reactors at the end of the projects, but now we tested the coating materials using the slides, glass slides, and for two different species, Chlora vulgaris and Atlea. And in near future, we will focus on also seawater species for this project. And the microalgal bioform structure and light penetration were determined after the 14 days of cultivations, and the biofilms on the coating classes were observed using the photometers. And to understand the biofilm structures on different coating classes, we utilized the fluorescent microscope to see the morphologic structure of the biofilms. And you can see that these figures, this is like coated slides, and this is without coated, non-coating materials. After 14 days, the non-coating slides are almost covered by the biofilms. But we would like to also see the light penetrations, and the two coatings material has a potential for light penetration when we compare with the non-coating materials. So it's the first results when we tested. The overall, it is observed that the pieces and particles adhere to the non-coated surface firmly. This is the slice without coating, non-coating at the left sides. And it is the coating material with 10.4. And this is also coating material with 12.2. And when we look the microscope images, we can see small dots here, but the penetration is very high when we compare the others. So we tested for the Atlea at the same time, and we observed the same coating material has a same potential as a anti-biofouling materials. So we focus on how can we increase the efficiency, especially the light penetration efficiency. So coating material is working now well, quite well, but it should be tested for different conditions. The next step will test with the outdoor cultivation systems, and not only slides, but also the all tubing reactor will be coated by the specific osmosyl coating materials, and then we will test it first of all in the lab scale, and then we would like to test outdoor. So the process of adhesion on the coating was examined by the means of fluorescent microscope. We observed, according to results, the 12.2 coating demonstrate the best light transmissions results for both microalgae species. Also, this project funded by TÜBİTAK. We would like to thank TÜBİTAK, and also we would like to thank Professor Berat Haznedaroğlu for supplying us in terms of pre-cultures as well as his knowledge on technical and scientific cultivations during the projects. If you have any question marks in your mind, I would like to answer that. So this is a new topic for all of us. We would like to increase the efficiency during the cultivation. Yep.
Thank you for the presentation. I wonder if you have carried out any comparative analysis for the economic application of scoring. Does it increase the installation cost up to 5%, 10%? I don't know.
Yeah, it's the main actual drawback for the system. The coating material is very expensive at the moment. So if we know the really good nanoparticle species, silicon nanoparticle species, then we would like to work on the decreases of the cost. Now it's really expensive.
I see. I might have missed, perhaps you already explained, but perhaps I missed it, but is that recording material designed by yourself? And
Coating material designed by us because of I have two colleagues from Çanakkale 18 Mart University from from chemical engineering departments. They focused on only the surface coating materials. So we invented, we also developed by ourselves.
excellent. Thank you.
You're welcome.
Dr. Küçüker, thank you so much. Great work. I know you haven't tested in your system, but the coating material is known for a while. Do we have a rough estimate? I mean, because even in the cleaning system, they are going to start worn out at some point, even with the water. Do we have a rough estimate how many cycles it can continue?
Actually, at the moment, in the laboratory scale, 3 times we can use the same slides. There is no harmful effect on the surface, but we will see on the full-scale applications near future. Then I can clearly say that. But this is the main drawbacks as well, because of the sunlights and some circulation in the reactors effects on the surface structure. We would like to see how many times it can be useful for cultivation.
Do we also know the thickness of what's the thickness of the coating?
Actually, it's very, very large, 0.01 millimeters at the moment, but the main perfectness is the contact angle. So we try to reach the contact angle like 10 or less than 10 degree. So, please.
Very nice work, and of course, much needed in the field. Can you tell me what was the main rationale when you designed your compound for coating? Was it really mainly the hydrophobicity properties, or did you think of any other physical-chemical properties?
Actually, in the future, first of all, we tested the hydrophobic situations, and near future, there are different kind of chemicals and superophobic under the waters as well. It will be tested, and maybe we will focus on the other physical properties of the coating material, maybe roughness or the flattening of the surface material, and it will be tested. It is like a pre-test of the first test setup, also first setup.
And this coating,
Yes, actually we also changing the other. For instance, this is the freshwater results, but the seawater is more harmful, and we will test it. In that time, maybe we should change the other properties, yes, the other components, maybe.
if I may ask just one practical question, so how do you actually physically do it? You know, when the tubular parts of the bioreactor are ready, or do you kind of premix it and then?
There's a specific, actually also we invented that material, like there is like a nozzle 360 degree pressing of the coating materials and pass through the inside of the reactors. And then also the other method is deep coating method. But in that time, it is really difficult because of we consume a lot coating material because of inside and outside coated if we use deep coating. So our aim to coating inside of the tubular reactor. So the special instrument was invented by us.
But as we heard today, the outside is also important because it is, has to be cleaned, right?
Yeah,
And that's energy expenditure, isn't it?
yeah.
So you really want to have it coated on both sides, I presume. Right?
Maybe in that time we maybe use a like less expensive material for the outside. So this is maybe second step of the research or further step of the research. Yeah.
Very, very important work.
Yeah.
Thank you. Congratulations.
You're welcome. Okay. Thank you very much.
Thank you for your presentation, Dr. Mehmet Ali Küçüker. Now our conference gonna continue with Snaz Albaki from the Islamic Azad University by the title of A Novel Hybrid Photobioreactor for Carbon Dioxide Mitigation via Chlorella vulgaris. The stage is yours.
Hello again. It's actually me again. This is Sina, and well, I wore glasses. Maybe this might be a little bit different for you. So I don't know. So okay. So this specific project that today I would like to talk about is a novel hybrid photobioreactor for CO2 mitigation by Chlorella vulgaris. What I can guarantee this time would be a little bit different because we just, passed the borders of the experimental thing in the laboratory, and for this one we already have a product, so maybe this can help a little bit. This specific project has been conducted under the supervision of Dr. Farshid Pajumshariati and Dr. Omid Tavakkoli, the contribution of the universities of Islamic Azad University of Tehran and also the University of Tehran. Yeah, I can work it. Okay. So at the very first beginning, the content that I would like to talk about is a little bit introduction about the greenhouse gases problems and solutions, and we're going to specify it with the CO2 capture. Then we're going to move to the materials and methods, talking about the microalgae, the one that we specifically used, and the photobioreactor design and the novelty that it might have. And the results and discussion will specifically related to the CO2 and nitrogen phosphate removal. And for the conclusion and the future, we're going to talk about the products that in the future or right now we can see the result of it. For the beginning, I can say that the impact of greenhouse gases dispersion improperly into the atmosphere is pretty obvious to all of us. The very common visible thing is the global warming that we can see nowadays. As we all know, according to the research conducted by reputable professors, the most, I can say, highlight 3 ways that we can mitigate this or control this greenhouse gas dispersion, and will be categorized on number 1, saving natural sources by decreasing the amount of electricity used, and number 2 is the direct way by cutting down the main sources of greenhouse dispersion, and also the third way is the indirect way by capturing the CO2. The thing that we can do on capturing the CO2 will be categorized in 2 phases. The first one is chemical, and the second one is biological. That we're going to stick with the second one and using microalgae. The thing that we have done in this project specifically, as it is clear in the picture, is the design that we have had on this photobioreactor, that we're going to talk about the application of it on CO2 removal and also nitrate of phosphate removal, or better to call it CO2 capture. The thing that matter in the design is that by adding different elements or baffles into the reactor, we change the circulation of it. And as we all know, photobioreactor is one of the main factors in using microalgae in different industries. That will be categorized into sections of open systems and closed systems. So this is actually one of the closed ones. In the material method section about the microalgae, we all know that microalgae are completely valuable products based on the products that they use, renewable energy actually application that they have, and also the environment that we can call it sustainable. So microalgae can be a promising kind of solution for CO2 mitigation or maybe saving the future. The whole process is the combination, when we are talking about design actually, the whole process is the combination of fluid dynamic, light transfer, and the reaction. If I want to talk about the things that we have done or the parameters that we have conducted is that the light dark cycle that we have chosen was 24/7, 24 light and 0 dark. And this one has been chosen according to the research conducted by our members in the laboratory that have been, that actually we categorized and tested 3 light dark cycle of 12/12, 24/0, and also 16/8. And this one was the best, specifically in nitrate and phosphate removal. So this is the chosen one. The pH remained constant in actually 8, and the light intensity is 3,500 lux. And also the temperature was the room temperature of 25 degrees. In the case of photobioreactor, the challenges in designing photobioreactor are vary type to type and based on the application. But in here, we narrow them down to 5 main groups. The first one is the cell movements in the light accessibility to cells and actually liquid and gas interface. And the next one is shear stress, mass transfer, and concentration distribution. And more specifically about the design that we have in here is the elements that we have settled into the photobioreactor, and that till actually for the next page I'm going to describe it a little bit more. As you can see, the whole process is using 3 different photobioreactors. The first one, okay, I'm going to use this one now. Yeah. The first one that has been nominated as the control group has no baffles, nothing inside. We can call it kind of like a bubble reactor. And the second one, which will be the airlift type, is using the 2 baffles for having a better circulation of the water and will kind of make the gas phase into the liquid phase retain time higher. And the last one is using the U-shaped baffle on the top, on the cap, as a cap. And make the circulation into the photobioreactor higher. All these 3 photobioreactors have been run simultaneously, and the results is that the CO2 capture, as the result shows, in the novel photobioreactor that has a different design is higher and, well, more promising. And the CO2 removal was investigated in this study, and the amount of CO2 removal is measured and analyzed terms during each period of the microalgae cultivation. OD dry weight and fluid retention time in the photobioreactor was determined after examining the CO2 removal process. And the whole scenario is the one that I described as 3 stages of the reactors and the comparison of each. Okay. As it is clear in the CO2 concentration in this result, the purple one is the bubble photobioreactor, or I can call it as the control group, but it's better not to call it the bubble. And the red one is the airlift, and the novel one is the PBR. The other parts and parameters remained the same as each other to just compare the design effect on removal. And as it is clear, well, the novel PBR performed better in removing the, or actually in CO2 concentration. And also in CO2 biofixation rate on day 7, again, novel PBR performed better. However, in some categories, it was obvious that the bubble reactor was performing better than the airlift one. And well, this is the experiment, so that's what it shows. But the whole process is showing that the circulation of the water and causing the retain time of the CO2 or air into the liquid phase increased the removal. The other result or the other phase of this study was using the cholera vulgaris microalgae as the second treatment of the wastewater. First, we performed it with sludge, and then we used the permanate of the sludge treatment through the microalgae. As you can see, the microorganism is cholera vulgaris. The culture medium was BG11, and condition is clear over the as pH, temperature, and also light intensity. And the wastewater COT, nitrate, and phosphate is as shown. The whole project was comparing three different stages of using only sludge, using only microalgae, and also using the combination of both strategies. And as it is expected, the combination strategy for treatment was actually better. And as you can see on the CO2 removal, well, the mixed culture performed better. However, the activated sludge application was also better than microalgae. And in phosphate removal and also nitrate removal, the mixed culture was obviously better, and well, considerably using microalgae was performing better. And actually all these experiments were in bench scale and laboratory scale, so they're not industrialized. So that's the results are showing. Is it possible to play the video? Do we have any technical support? I don't know if this is working specifically. Sorry. Okay, very good. To the end, actually, the Mother Earth is the only thing that we all have in common. And we must make a great effort to transfer it to the next generation safe. Microalgae can be one of the most promising solutions, but we know that these valuable creatures are facing with billions of difficulties and maybe problems. But what if we could change the metabolism of microalgae the way that we expect, so we can increase the CO2 removal, CO2 capture, or maybe in the future everything can change through these biological changes. We know that large industries like agriculture, aquaculture, and animal husbandry are among the most important investment in the future, which actually are associated with high cost and high energy consumption. Based on more than 20 published articles, the technology of advanced chelate compounds has shown that it can increase the yield from 20 to 200% in agricultural industries and increases the surface of leaves. In one of the interesting published articles on this technology, a considerable amount of absorption, the considerable amount of increase in absorption of carbon dioxide has been performed by trees of pistachio in harsh conditions in Iran. Which we all know that pistachios are type C3 plants, so the condition is kind of like a dry weather and well, challenging. This technology works best on the unique features of metabolism optimizer, and by using this technology on microalgae, which we know that they are one of the most, I don't know, reputable, highlighted one in agriculture products as fertilizers or animal feed supplements. Maybe can by optimizing the metabolism of these little creatures, not only we can reach the efficiency of the industries by increasing the products, but it also is a possible way to guarantee in biomass production and, well, saving the future. The technology that I'm talking about, known as chelate compound, is a technology that Sodur Ahrashar knowledge-based company owns. And it has been seen the application of it on different types of plants. And also, nowadays they are having like 17 different fertilizers, biofertilizers and nanofertilizers, and more than like 5 or 6 animal feed supplements. And the multidisciplinary use of this technology can be quite interesting in microalgae application. And the technology that we're talking about, the chelate compounds, has been patented in United States Patent and Trademark Office and European Patent Office under the name of the CEO of the company, Mr. Mohammad Hassan Nazaran, which can be promising in the microalgae industry world. This is all that I want to talk about, and as you can see in here, the two products that we are working on is in the fishery industry is bonza fish, that we are using the microalgae and also this specific technology of chelate compounds in creating the fish feed supplements for marine warm water fish and also cold water fish. And also it includes different type of feed supplements like livestock and other industries. And the other category is the fertilizers that we are using recently. Well, there is a vision there that it can be kind of like quite promising in the future by increasing the yield from 20 to 200%. And here is our team. Well, that one is me, Sina, and Dr. Farshid Pajunshariati, and Dr. Umid Tawakkoli. And the investors are Sodor Arashad Company, Islamic Azad University Science and Research Branch, and University of Tehran. Thank you so much, and I promise this is the last time you are seeing me here. Yeah, thank you.
So I didn't ask for the microphone.
Sure.
So we can
Yeah,
shut
no,
it
that's
off.
okay.
Thank you very much for your talk. This is a bit of a statement and a question to you, because there is obviously a big difference between CO2 capture, sequestration, and CO2 utilization.
Yeah.
That algae need CO2 is no doubt, and
Yeah.
even more than atmospheric CO2. You can do it with 2%, 4%, maybe even with 10%.
Yeah.
But there is an upper limit.
Mm-hmm.
But to my knowledge, at least on the European point of view, the discussion on CO2 capture and storage and sequestration has been finished already 10 years ago, because
Yeah.
people calculated that that is impossible.
That's so true.
Because we calculated that a middle-sized German coal power plant
Yeah.
would need hundreds of square kilometers of algae plant to just sequestrate one of these power plants.
Yeah.
So I would ask you, what are you calculating with your new systems?
Sure.
And if it's not too much, you should be careful when you talk about capturing.
Yeah, sure. That is totally true, and actually it is a considerable situation when we are talking about capturing thing, because it's like a big word, and we are all aware of it. But the thing that we're doing here, as I said on the previous presentation, it is a very small part of a project, and the whole concept of the project is just trying to investigate on every single part of it to just collect data and everything possible. And the main project, or the main reason that we are using these things is for the pigments and technically astaxanthin or other pigments that we are using, like beta-carotene, because these are kind of like things that in Iran we are challenging with, like beta-carotene and other pigments, that we need to import them all to our country. So these are kind of necessary things in our country. But in case of capture, yes, that's right, and that's totally true. We should be aware of what word we're using specifically, and this is what we are doing as just a very small part of the project, and that's the name that maybe we are supposed to use it, you know. So that's how it works. Yeah. Sure. Is that okay? Yeah, please.
Thanks for the great second
Thank you.
presentation. Actually, I want to comment on the previous question.
Sure.
You have so many deserts, actually, right?
Yep.
In the like Middle East.
Middle of Iran, yeah.
Yeah, many deserts, and I heard of some projects. Yeah, maybe they are not very like solid projects, but some projects on the desert they produce algae for carbon capture. I don't know the like limits, but I think maybe it's not feasible in Europe, but maybe a big desert with the like high
we do have, but we cannot say that it is kind of like acceptable to, because there are lots of deserts and lots of areas, empty areas, we can use it. No, of course not, because we are facing with a harsh condition on those specific deserts that barely creatures can live. So, and it's going to be like very time consuming, and it is really not sufficient or cost effective, because when we are talking about the industries and when we pass through the industry phase of everything, like feasibility study and whatever else, we are not able to just consider the results or the locations or whatever else. Every single thing matters, and we need to consider every single part. That is why technically the application of microalgae in Iran is kind of like a dream, dream because maybe it is not possible to have those applications in industrial scales in our country. However, there are some specific species in Iran, like Donalila salina. We have lots of lakes and everything else in Iran that they are really, well, interesting and usable, but unfortunately, no action can happen because of the thing that I just mentioned. But yes, as you said, it's totally true. We do have some condition to grow algae and everything else, but it's not just about the area or land or places. We do need facilities, we do need equipments and everything else. That if you just collect all this information together, we need to stick with the lab scale and like bench scale right now at the moment, and then we talk about something more than that. Yeah, thank you.
Thank you, Sina. So your reactor system, are we still doing better carbon dioxide capture?
Compared to the other two.
Yeah, but you haven't mentioned the pH level, so you must have more carbonic acid in there. How low the pH levels go in the system?
Well, the pH, we try to keep the pH level in a kind of like remain in this whole process. We kept it on 8 or 8.2 kind of thing. And we just wanted to know about the difference in designing. So that's why we compared only those 3 reactors together.
So does that mean that you have a buffered system
Yeah,
compared to usual?
Yes.
So you have more buffers in the media?
Yeah.
Okay. My second small question is, you got quite a few, CO2 removal, although you're pushing CO2. Were they sort of like uni-algal systems? Or you mentioned algal only and also mixed cultures, but
in the wastewater.
Still a mixotrophic system? You're pushing carbon dioxide?
No. Technically, you're talking about the wastewater treatment part, or?
Yeah, wastewater part.
Yeah, for the wastewater part, we haven't considered any of, and we didn't use the wastewater and compare the CO2 kind of like capturing or
No, I
utilizing.
understand that. My question is on that part.
Okay, sure.
You had quite a bit of COD removal in algae-only systems, but
Yeah.
that was autotrophic, not
It was no autotrophic, not mixotrophic.
But the system was still uni-algal.
It was kind of like we used actually the pre-treated wastewater of the sludge, and then we used it, the synthesized one, for using it via microalgae removal or microalgae application on it.
I'll ask that. I still have some questions.
Sure.
Thank you.
Okay, I guess that's it. Yeah, thank you.
Thank you for inspiring presentations, Algae. And now we are connected to Dr. Işılay Temizdanyar from the Turkish Marine Research Foundation by the title of Algal Blooms, Marine Mammals and Multidisciplinary Preventive and Protective Perspectives. Could we hear you?
Hello everyone, can you hear me well?
Yeah, we can hear you properly.
Presentation, can you see
Yeah,
it?
we can see it.
Good.
Okay, we are ready to listen you. Thank you.
Thank you. First of all, I would like to say that my thoughts are with everyone who has been affected the disaster in Turkey. My name is Işılay Temizdanyar. I'm a member of Turkish Marine Research Foundation, and I'm a veterinary pathologist in a different area. And I'm currently doing my postdoctoral research on marine mammals in Italy's Padua University as a gentleman scholar. So today I will try to take you from the microfauna to megafauna of our oceans. Yesterday, Professor Vera already covered algal blooms in detail, and Professor Smith covered algal metabolism, so they eased my work. But let's say some words about the algal blooms. As we know that they are the overgrowths of algae in water, and harmful ones affect both human, animal, and also the environmental health. And they affect local economies by producing the harmful toxins in fresh and salt water. They can be observed in marine, estuarine, and freshwater habitats that we can observe marine mammals. Their increased global distribution, intensity, and occurrence over the past few decades are emerging topic for us in terms of marine mammals health. Out of 1,000 algae species, there are around 200 species causes hubs, and that can be transferred to the food web and can affect multiple trophic levels. And as the marine mammals are the top predators in the food web, it is important to know the way of exposure, type of the algae, the effects of the biotoxin or the algae on marine mammals, ecosystems, habitats, and their own bodies. Marine mammals, as I said, they are the top predators in their ecosystem. We have cetaceans, which means dolphins and whales, sirenians, manatees and dugongs, pinnipeds, which are the seals, sea lions, and walruses. And also we have otters and polar bears, which are also considered as marine mammals. So they have important role for their ecosystems, their food webs, biodiversity, and of course the marine habitats. And they are sentinel species for ocean health, as they live too long lifespans and they share the ecosystems. This is the 20 of the cetaceans that we have in all Mediterranean basin, including the Black Sea. There are 78 cetaceans all around the world, but we have 20 of them in Mediterranean. In Turkey, we can see 12 of them. And also we have, from the Pinnipedia, we have our precious Mediterranean monk seal, which is endangered. And here you can see the harbor porpoises in Bosphorus. Of course, we all know that marine mammals are trending, and sometimes they are called unusual mass mortality event that we try to understand why they are trending in high numbers. There are some reasons like diseases, fisheries interactions and bycatch, habitat degradation, overfishing, deliberate killing, traumas including the acoustic traumas or ship strikes, malnutrition, environmental degradation and pollution, and harmful algal blooms are also listed as one of the reasons for marine mammals deaths all around the globe. Harmful algal blooms can directly affect the marine mammals in lethal way or sublethal toxicity for adults, youngs, and also they can affect the babies in utero. And also there are the indirect effects like hypoxia, changes in food quantity and quality, therefore stress and susceptibility to other diseases. So we have to take in multiple angles the problem. And there are the other important factors, of course, the exposure route, the dose that they have. The species, both the algae species and also marine mammal species that affected, their life stage, their life history. We have to know their well-known about their ecosystem, temporal, spatial synchrony, acute and chronic exposure, whichever is the problem at the time, and interactions with also other stresses. So we have, as many of you already know, we have the One Health approach, which means good environmental health, good animal health, and human health should be considered as one. and we have to know better. It is important for us to better understand the effect of algal biotoxins on marine mammals and develop multidisciplinary solutions. From the veterinary point of view, it is always necessary for us to have sustainable data to better understand the algal bloom events and their effects on marine mammals, and evidences that we can find in marine mammals, because we can sample the animal, but the sampling life, shelter life of the sample, sampling time, they will be all related with the type of the biotoxin or the different environment. Mainly brevetoxins, dinoflagellates, and diatoms are those mass mortality events, especially around the North America. There are the main cases that we know, but this is also another reasons why we don't know that much about that regions, because they have the, I would like to present this data from the NOAA Fisheries. It's a governmental organization, governmental institution, and they have been tracing the causes of unusual mortality events since 1991. And as you can see, 18% of the death reasons, the unusual mortality event reason, based on biotoxins. And they can trace the reasons since 1991, and in 1996 you can start to see biotoxins and the unusual mass mortality events caused by biotoxins, and then it is continued to be monitored. So this is important from biological perspective, of course, the environmental perspective, but also from the health perspective, to better understand, because sometimes they are the mixed problems, like there can be the morbillivirus infections around the area, which also cause mass mortality events, but the animals' immunity could be decreased because of the biotoxins. So we have to better understand those problems together in connection. So we have current methods for detection of harmful algal blooms, but also their effects on health. We have some diagnostic methods, but we need some fine-tuning. As I mentioned, we need to understand better the pathology of the problem. So all mixed approaches are also emerging ones are helpful for us to better understand. For algal blooms, there are physical, chemical, and biological control methods. All of them has some advantages and disadvantages, and environmental scientists trying to better understand ways to control harmful algal blooms. But from the One Health perspective, it has to be the monitoring, the detection, the control, all of them has to be linked so we can better understand our macro environments and micro environments. This is the slide that I usually use about protection of marine mammals, which requires well-developed bycatch and stranding networks, which means countries has to establish, especially if they are part of the ACOBAMS agreement to protect the stations. Our country is also part of the agreement since 2018. The countries have to have the well-developed bycatch and stranding networks, which means the countries also have to monitor the animals that are already died. But if they can reach, those are the animals that are so precious, even if they are died, so we can have understandable data about our oceans' health. Multi-interdisciplinary approaches are needed, and multi-country response and international collaboration is needed because of the marine mammals that are the creatures that does not know about the borders and they swim freely. So we need multi-country responses. And of course, we need citizen involvement because you cannot be here everywhere each and every time. Turkey has 8,333 kilometers long of coastal lines, so you cannot be there each and every time. But if you have the citizen involvement, if you have the coordination, you can always reach the animals that also life-stranded ones. So there could be hope for them too. National and international specialized instruments, organizations, and funds are needed, which is also common for controlling the harmful algal blooms. So we need the monitoring programs and early detection systems, management, control, and remediation strategies, but we have to do them in line and in close contact with One Health policies and their implementations in place, or we have to develop them together. So what to do next to protect marine mammals from the algal blooms and better understand their relationship? Of course, we need effective coordination, prevention, and management strategies and tools to evaluate multidisciplinary data, like the data on algal blooms and also marine mammal health, which are completely different. That they require different occupational scientific approach. But we need to establish national stranding networks or improve them or develop them. Establishment of early detection, monitoring, and response tools on harmful algal blooms, because we know that we have algal blooms in Mediterranean, in Black Sea, which are the vulnerable seas. And we have unusual mortality event in all Mediterranean basins. But we can still have some relations between algal biotoxins and marine mammals that we don't know yet. So we need interdisciplinary work. We need networks between those scientists, databases on algal research and also biotoxin research and also veterinary experts, marine biologists, and environmental scientists working in the same area. Before I conclude, I would like to, from all of you, see this photo and remember it all the time. When you have a life-stranded animal, you can easily help the animal by taking them in an area that you can provide shade, by a piece of thin cloth and keeping it moist all the time, and protected blowhole, which is the nose of the marine mammals, from dust, from sand, from water, so they can breathe easily. So you can also keep cool the fins, because they are the center for thermoregulation for them, and as they are out of the water, they cannot control their thermoregulation. So you can basically help life-stranded animals until the veterinarians or other helpful personnel can reach there. In Turkey, you can reach to Coast Guard, or in some places, the Coast Guard also directly linked with 112, the emergency phones. So we can have hope for all of them. And the audience in the room, if you will spend your weekend in Istanbul, take a Bosphorus tour. If you have, take your binoculars with you. You can always see a harbor porpoise or a bottlenose dolphin or a common dolphin in Bosphorus, which is a place that one of the best places in the world that you can see dolphins in their natural environment easily. So thank you for listening to me.
Thank you for your presentation, Dr. Işıl. If you have questions, I will direct them. Do we have questions? Yeah.
Thank you for this wonderful presentation that is, it's so important for the microalgal community to get out of their comfort zone and really see how, this amazing microalgal ecology can have a, strong effect on the mammals, marine mammals. So you mentioned in your presentation a point that is extremely important, I think seriously underdeveloped amongst the microalgal researchers community. It is the early detection of the HABs, the harmful algal blooms. So do you know of any initiatives? I know that there has been some call recently that obviously, I think it was called Biodiversa Plus or something like this, where, possibly there were some attempts to address this problem. Satellite imaging, again, totally underdeveloped. And I think we as microalgal scientists and those of microalgal scientists who are into photosynthesis can really help here, right? Especially with the prediction, early prediction of the microalgal blooms by, for example, using the specific signatures from various pigments that are very often specific for specific, algal, maybe not species, sometimes species, but mainly, algal taxonomic groups, right? So do you know of any sort of like global or at least European initiative that would try to combine this knowledge into the early detection of HABs?
As far as I know, UNESCO has some tools about the algal blooms. They providing some reports on global algal blooms. And also, European Union has some different regulations, and they under those regulations, there are some common databases trying to be established. Also for the satellite imaging, there are some tools, but as you said, they need to be still developed to detect which type of algal bloom is going on there, and how can we know about their distribution to predict the area to zone. As far as I know, based on my information, there are still, as you said, underdeveloped, but there are some common grounds like UNESCO, like EU's working groups, and probably it will be developed soon. Thank you for your question.
Right, thank you.
Thank you for the presentation. I really enjoyed it. I really wonder your personal opinion as a member of the Turkish Marine Research Foundation. The topic is that the algal blooms that we see in Antalya and Izmir, the Ulva lactuca. Is there any strategy to deal with that from your institution, or your personal opinion on that? What we can do about it?
For the algal blooms, as I'm a veterinary professional, for algal blooms, I can say that when you go into UDA's website, there are special studies for Marmara. There are also some special studies for Aegean and Mediterranean, and they are all published in the publishing part, but they are mainly done by the academicians all around Turkey. So as far as I know, there is a big team in Istanbul University, Faculty of Fisheries, and there are also other many universities in Aegean region, and also, as far as I know, ODTÜ, Middle East Technical University, has some studies on algal blooms.
Işıl Hocam, thank you so much for your talk. I also would like to ask your comment on one topic. So we know that the Mediterranean basin is warming up much quicker and much more than compared to the other seas in the world. Do you see changes in the HAB formations in recent years, especially after 21st century after the year 2000 on the Mediterranean basis?
Based on the reviews I checked before this presentation, there are some increasing signs, we can say. But you know, Mediterranean and also Black Sea, Marmara, they are so vulnerable environment, but they are also so precious with its all habitats. And this is not the only algal bloom part. There is the climate change, there is the invasive species. Maybe we also still don't know the relation between the micro algae with the invasive species. So we need to better understand. We have to collaborate all around Mediterranean. We have to collaborate. It is vulnerable to climate change. It is vulnerable to thermal change, and of course, it will cause more harmful algal blooms in the real near future, as far as I can understand from my veterinary point of view.
Thank you. One other question that I would like to again ask for your comment. So our anthropogenic events, just like the mucilage in the Marmara, we are putting so much nutrients in our closed systems, again including the Mediterranean. So we are actually causing non-harmful algal blooms become increase in number artificially, and they become harbors for pathogens, viruses. I haven't seen any study their impact on the mammal system. Do you see, for example, was there any impact from mucilage on the mammals? We know small fishes, but I mean, we are causing as humans, unfortunately, our anthropogenic pollutions, causing non-harmful algae to turn into, reservoirs for pathogens, basically. Can you comment on those impacts, maybe?
But even they are considered as non-harmful. They have effects in our systems, like they have effects on fishery production, their reproduction. So if there is a problem with the even it is non-harmful algae that are produced at all, then there's a problem with the fish reproduction. And then there is a problem with the marine mammals who are dependent on those fish to, prey, to reproduce themselves. And, when the marine mammals are affected, they have long lifespans, but at maturity in long time, they can give birth for after 1 year. So we have many other problems like stepping up the stairs. We have first the non-harmful algal blooms, and when the ecosystem is affected, it's going up and up.
Thank you. Thanks so much.
Don't have more questions, I think. Thank you for your presentation, Dr. Işık. We really appreciate it. Take care of yourself. Now, we will continue with Cemil Koyunoğlu. Since we have switched the places of Dila Hocaoğlu and Cemil Koyunoğlu yesterday, we will continue with Cemil Koyunoğlu from the Yalova University. His speech title is a review on the catalyst effect of bentonite in biofuel production from algae. The stage is yours.
Hello everyone. Today I want to talk about how effects of bentonite as a mineral material for the production of fuel from algae. So let's start why bentonite is useful for the production of fuel. I want to explain theoretically, first of all. If you imagine a bentonite, it's a chemical structure of silica mainly, so it means it can easily combine with oxygen. So during the production of algae with biological reactions, so I will explain on the chlorella based species. There are some studies based on it. The first article is based on the harvesting effect of bentonite. So the conditions of experimental is, I can say, the pH range between 4 and 10, and the flow of the CPAM is 1 milligram per liter. So they take about 3 minutes. So after this experiment, they used lots of kind of mineral, but the point here is the bentonite increase the efficiency of the oil from algae. The second article is mainly based on the chlorella vulgaris species. The effect of the bentonite they used in this study. So they investigated that the increase of the bentonite ratio in the media of the cultivation. So after the experimental results, they found the peroxides activates is increased. The third one is the same species based. So they focused on the only plasticizer adsorption, because the bentonite is useful for adsorption reaction in the chemical industry. They use the surface agents mainly in the chemical industry as well. So in the study, they used a kinetic method to see how bentonite affects of the removal of the BPA molecules in the medium. So there are some blending options in the experimental sections. In the first step, they blend with microalgae and lignin. So it means they have high value carbon. So after the reaction, the ratio of hydrogen and carbon increased. It means the silica element in the bentonite combined with the oxygen. So the results, the remains hydrogen and carbon ratio will increase. In the same subject and the same step, so the fifth article, fifth study, we can say if bentonite usage in the also some elements composition medium in the same chemical behavior as I can say absorption. So in the, they characterize only the catalyzed effects of the surface. So they used UV visible, NMR, LCMS, and FTIR. So they can see after the some chemical mixture. So bentonite structure can easily transfer some anions to the remains solution. I guess in the last article, but there are lots of study based on the chlorella species. I don't know why another species they couldn't study. So in this study, the chlorella vulgaris algae cells, they used bentonite as a chemical, as a catalyst. So in the absorption based, the bentonite can remove chromium ions from solutions. So it can increase the zeta potential of the solvent. So as you may know, the bentonite and another mineral kind, boron minerals, can affect on the solution to get balance in the minerals in the solvent. So they can put lots of groups in together in the solution base, so the chemical reaction can easily occur in the medium. The reference of the chemical behavior of the bentonite is a zeta potential. So if you can observe zeta potential of any solution in the algae medium, so you can see the homogeneity of the chemicals. So it can say you can increase oil production in the algae. So in theory, as I mentioned before, so the reaction mechanism of the bentonite can benefit in the medium homogeneity of the ions. So in the study, you can see in the ferrite composite in the solution can be removed by the bentonite, so it can easily provide a homogeneity of the solution. If I have time. Okay. So if you want to produce more fuel from the algae, so you cannot use a high value of the bentonite. There are some limitations. So if you want to observe the limitation of the bentonite, you can make the Langmuir isotherm-based study. So you have to find the limit solution of the bentonite. In this study, they used some experimental design methods called as the Taguchi. So they can use, they used about 10% of catalyzed concentration. So there are also another minerals in the bentonite called the magnesium oxides. So it is very important mineral to absorb the oxygen in the algae medium solution, so it can increase the yield of the oil content. It means the maximum energy content. In this study, they produced the biochar with energy intensity of the 21 and 26 megajoule per kilogram. So it can same value as the coal value. So it is very important study. So in this study, they used the bentonite and the the raw material is the waste, green waste and sewage sludge. So they put each of raw material as a different phase, different step of the experimental. So they produced biocrude, gaseous product, solid and water phase product. So in this study, it is the chemical conversion of the algae. So they found that the Chlorella vulgaris usage, there is a synergistic effects of bentonite if you use in the hydrothermal liquefaction. This is a different chemical conversion method to produce fuel. So if you use the nanoparticle size catalyzed, so you can change also the efficiency of the harvesting of the algae. There is a study you can see in the slide. So if you use the thermal composition, thermal conversion, the particle in the nanoparticle size can benefit to put the unwanted materials, the kind some ferrous materials. So if you put magnetic nanoparticles with another particles in the medium, so you can increase the oil production. So there are lots of observation in the study. If you want, I can send by email with these calculations on the observations as well. So if you use the bentonite with chlorella harvesting, in this study they used the Langmuir kinetic method. So they found the second order reaction can increase the chlorella harvesting because they remove some materials in the medium. So they found some results of some specific temperature, some operation temperature. So they found the bioabsorption capacities increased. Thank you for listening. If you have questions, I will be glad to answer. Thank you.
Thank you for presentation, Cemil Koyunoğlu. Before we are leaving the hall, I needed to announce two things, but the first, I will leave the last word to the chairman and homeowner, Berat Haznedaroğlu. Thank you.
Thank you, guys. Okay, we're almost over. Since we stole a little bit of time from our poster presenters, I would like to mention that we will keep the poster hall open another hour so that we can also, say our farewells for those that might be leaving and for final chats. So I just would like to take a few moments to thank a few people. The first one, of course, goes to you. It was really, really hard, I know, for everybody to arrive in Istanbul, especially in this week, some for the weather and of course the tragic event of earthquake. So the big thank you goes to all audience. Thank you so much for being with us for the last 2 days. I think we covered really, really great Nexus talks since yesterday. We touched base in both water, food and energy concepts. Our hope still, just like Luisa's talked today, we hope that the algal biomass will be will have a significant role I think that's the best way to put it, in the toolbox that we will have to address the major issues of the humanity for the next 50 years at least. I would like to thank our keynote speakers. Thank you so much for accepting our invitation at short notice and then coming to Istanbul in their busy schedule. Yagut, Olaf, and Vitor, I would like to thank you individually. One thank goes to our funding sponsors, the European Commission, and also the Turkish Ministry of Industry for making the independent project live for the last year. And also the Turkish Airlines for our transportation sponsor, and also the Polat Makina. I would like to thank my organization team. Thanks so much. If you could guys just, stand up for a second so that we could thank you properly. You guys did a tremendous job. Synapse selection as well. Thank you so much, guys. We have two friends in our direct translation cabin. They did a tremendous job, Burcu and Deniz. It is so hard to do real-time translation on technical terms. We didn't give them anything. Everything was spontaneous because, all the presentations were uploaded as we speak. So that they did a great job. Thank you so much, guys. For those that are staying for the biorefinery visit tomorrow, just a reminder, it used to be 9 o'clock departure. It will be 10 o'clock departure tomorrow, right in front of the hotel. So if you could be here about 15 to 10, it would be great. Our friends will be, I'm not gonna be here, I'll be at the biorefinery. So if you could just follow the organizational, team tomorrow, it would be great. Again, thank you so much for being in here. It means a lot. This event became sort of like a big morale for all of us, having you guys in here. So thank you so much. Bye-bye.